Hi, AP Psychology students, welcome back. Today we are continuing our discussion of neurons and we are moving into focusing on their communication. So this aligns with College Board objective, explain how the basic process of neural transmission is related to behavior and mental processes. So in order to get at this objective, we're going to focus on two essential questions. The first is, what are the steps of neural transmission within and between neurons? And then how do disruptions to these steps lead to issues? So when we talk about neuron communication, it's important to know that there's two kind of stages. So the first one is communication within the neuron, and this is generally referred to as neural transmission. So it's the idea of how the message moves from the dendrites, right, who are listening, right, through the axon to the terminal button or branches, or the axon terminal, the terminal at the end of the neuron. Then the next step, or the next stage, is the communication between neurons. So this then includes all the things that happen from one neuron, right, the axon terminal of one neuron, to the dendrites of the next. And what are the steps that it takes for the message to actually move from one to the next neuron. Okay, so we're going to focus first on communication within one neuron. So just some basics on the way that neurons work is that neurons move their messages down the axon, right, by firing an impulse. And this impulse is called an action potential. This is just a brief electrical charge that travels all the way down the axon, and with it, that's the message moving. Neurons generate their electricity for this charge from chemical events that happen to the neuron. So if you take a look at this picture over here, you will see that when you're looking at either a myelinated or non-myelinated neuron, that you have the positive charges outside and negative charges inside, right? Now this is where the neuron is at normally, and what we know about electricity and the way charges work is that because there is a negatively charged, because the axon is negatively charged, it attracts those positively charged ions, and that movement of ions in and out of the cell is what creates the electrical charge and moves it all the way down the axon. And as you can see, it's slightly different with myelinated neurons. The action potential kind of like jumps from space to space, and with non-myelinated, it just continues in one big kind of message. Okay, so there are a few kind of rules that we follow in neural transmission. So the first rule is that in order for a neuron to start, in order for a neuron to send this message, for the action potential to get going, for ions to flow in and out, it has to reach what we call a threshold. So a threshold is just the minimum amount of stimulation that needs to happen. A way that I want you to think about this is like, okay, so if we have our dendrites and they're listening, it would be like maybe one dendrite is listening, two dendrites are listening, they're both listening. Okay, that's not enough. Now we have 3, 4, 5 dendrites. Okay, and the message sends, right? So they need a minimum amount of stimulation. If you look at this graph over here, what I was trying to demonstrate with my fingers is this green section right here, where it's like, okay, we have some stimulation, then not enough, then more, then not enough, then where, and then woo, we go. And once stimulation happens, it happens. That leads us to the next point, which is that neurons are either all or none. So they don't partially fire. They either fire or they don't fire. If there's not enough stimulation, they don't fire. If there is enough, it goes. Sometimes students are like, okay, well then how come some messages are stronger than others? It's not about like faster or slower neuron firing for the strength of a message. It's more about the number of neurons that fire. So for example, the difference between like a big smack on the back versus like a light kind of pat on the shoulder is that a smack on the back would require more neurons and they would fire more often than like a kind of nice kind of soft touch on the shoulder. Okay, so once we've hit that threshold, right, that's when the kind of magic starts to happen and the movement of the action potential goes down the neuron. So while we are waiting for that threshold to be met, we are inside what's called the resting phase. So there are 3 general phases of neuron communication. While we are waiting for that threshold to be met, we are in that resting phase. This is where, as you can see, there's a negative state inside the axon, and outside the axon, in the fluid surrounding the axon, is a positive state, right? And as you can see in this picture, we have that stimulus that's happening. It's listening, right, and waiting for the threshold to be reached. And once that threshold is reached, that's when the next phase begins. So a little reminder, resting phase is positive outside, negative inside. Okay, so once we've hit that threshold, then the depolarization phase begins. And what happens is that there are these gates, right, that open and close on the axon. And what happens is when the threshold is reached, right, the gates open and those positively charged ions flood into the cell. And as they flood into the cell, then that actually stimulates the next gates to open and flood into the cell, and the next gates to open and flood in the cell. And this happens all the way down the axon until it reaches the terminal, axon terminal. So I want you to think about this as like dominoes, right? That one triggers the other, triggers the other, triggers the other. If you take a look at this diagram here, you see that we have these positive ions. They are sodium ions, Na, but you don't need to know them. You don't need to know what kind of ion they are, but positive ions are floating in. And then what happens is that then these positive ions that are floating in trigger these gates to open, and then they switch back, and the next part of the neuron, the positive ions flow in, and then it moves to the next one, the next one, the next one, all the way down, right? So it's a process that moves down the axon. It's not that the whole axon changes at one time, right? Dominoes all the way down. This is like another example, kind of close-up of what it looks like for a myelinated neuron. So you notice here we have a depolarized membrane, right? Positive ions have gone in. And this section, the next section, is at resting rate. But what will happen is that this depolarized membrane will trigger this one to activate, and then the next one to activate, and then the next one to activate. So it jumps down the axon. Okay, so after the message has left that section of the neuron, right, after it has left and jumped or moved on, that part of the neuron goes through what we call a refractory period. And this is like a resting period where the neuron kind of has to like reorganize itself, make sure the neurons, or make sure the ions are all on the outside, all on the inside, that the correct charge is all there and ready to go before it can send again. While this is happening, no new messages can be sent. And it's very quick, right? It doesn't like take a long time, but the neuron needs to rest before it can send again. If you think of it kind of like a toilet, this helps me. So you can't like flush a toilet when right away again, right? It needs to fill up before you can flush it again. And your neuron is the same way. And that period where it's filling up would be the refractory period. Okay, so now the message has reached the end of the neuron. And it's moved through the dendrites. Listen, it did the wave with the axon, right? And now it's in the terminal branches. And as we know, we talked about in class, what happens in the terminal branches is it's got to spit, right? So now what happens what is that really that spit process, right? So the first thing that happens is that the terminals, right, our axon terminals, hold neurotransmitters. And you can see that in the graphic right here, right? Now you'll also see that this graphic labels the like little kind of like sac that the neurotransmitters are in, called vesicles. You can, it's a correct terminology, but as far as we're concerned in AP Psych, just knowing that the neurotransmitters are inside the terminal buttons or branches or axon terminal is all you have to know. Now what happens is that these neurotransmitters move and they actually release out of the axon terminal. And that's that, right? That's spit. Now, as you can see, when they release, right, they send across this space called the synapse. And that's the next step. So the neurotransmitters travel across the synapse. It's a teeny tiny little gap. And once they get across the synapse, they then attach to the receptor sites of the next neuron, the receiving neuron, right? And they fit kind of like a puzzle piece, right? So every neurotransmitter has a unique shape, and dendrites have unique shapes. And so certain dendrites only have certain neurotransmitters, and they kind of fit like a lock and a key or a puzzle piece, okay? So you see here, again, just as a reminder, the neurotransmitters are in the axon terminal. They move to the edge, they release, they float across the synapse, boop, 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 and they kind of lock into the receptor sites of the next dendrite. Okay, so while this is happening, the receiving neuron, right, all of these little neurotransmitters are getting locked in. This receiving neuron is like listening, right? And the idea is that once enough of the neurotransmitters have locked in, a threshold will reach and the signal will continue. Now, that's not the end, because what happens is that the neurotransmitters who just did the communicating, right, they locked in to the receptors, they now get vacuumed back up into the cell, right? They get vacuumed back up into the terminal. And as you can see, this process is right here. This process is illustrated by the green arrows. It's called reuptake. So they get vacuumed up through these little transporter vacuums back up into the cell where they get reutilized. Now, you can also see from the blue arrows that not all of the neurotransmitters like make it back into the cell. Some of them like kind of get disintegrated in the synapse, and there's other things that can happen to them, but a large majority of them get like sucked back up through a process of reuptake, right? That vacuum process, reuptake. Okay, so that's how neurotransmission is supposed to work, and when it works properly, messages continue to send in the nervous system to their end place. However, disruptions can happen where something goes wrong. So we already talked about this a little bit. An example of disruptions happening at any stage would be disruptions happening where, like, within the neuron. So in this example, you have damage to the myelin sheath, which you see right here, right? And that damage causes a slowed or stopped message to travel down the axon, which then can lead to problems with muscle movement, tiredness, etc. And that's a disorder we know as multiple sclerosis. Now, another example of where disruptions can happen would be at the receptor sites. So I release my neurotransmitters, right? And then I have my dendrites listening. And there are some disorders where dendrite receptors are blocked and they cannot receive any messages. So if you look at this picture right here, you see these little antibodies that are created by your own body. It's an autoimmune disease. Block the receptors so that the neurotransmitters, in this case acetylcholine, which we'll talk about in the next video, can't connect when it's being blocked, right? And this disorder, in this case, where you have antibodies blocking acetylcholine, it really impacts the muscle droopiness of the muscles or process of the muscles, and this is called myasthenia gravis. And then another way that a disruption can happen is actually from an undersupply of neurotransmitters. So this terminal, right, when it releases, maybe there's not enough neurotransmitters that it releases in order to stimulate the next cell. So if you kind of look here at this picture, you see this would be a normal kind of situation where you see you have two receptors and many more being sent, and one receptor and many more being sent. And then when someone maybe has an undersupply, that can happen in depression, you see that there are two receptors but only two being sent, and like one and one, so there's no extra kind of message that's available to really make sure that everything is sending and responding properly. All right, that's all for now, AP Psychos. Remember to stop here and make sure you can answer these essential questions. And remember, psychology is flippin' awesome.